This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 197, recorded on April 25th, 2019. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there.
How are you doing?
I'm doing fine. Life is easy in San Diego, as you know.
It's starting to get warm here. We're in the 60s.
We had a cool winter, surprisingly so. I don't say cold by any means, but cooler than usual.
Yeah. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
Actually, you're not in Ann Arbor. You're in Sarasota, right?
I am enjoying a little holiday, a little work holiday.
Nice.
It's lovely.
Good for you.
Work hard, play hard. My first boss, Sam Silverstein, who you know, Vincent.
I do.
Said that you get more work done in 11 months than you do in 12.
So he advocating take a month off a year?
Well, not necessarily continuously, but just emphasizing that you burn out. You can't think well if you're always on.
And
There's
also,
a lot to be said about that.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How are you doing, Michael?
I'm doing well.
We haven't heard each other in a month. We've had a weird schedule lately, but I'm hoping we can get back on a twice-a-month schedule as soon as this semester is over. It's more fun that way to hear you guys twice a month. Don't you think?
It is. It is. Absolutely.
Yeah.
I miss you guys.
Yeah.
Yeah, me too. But you've kept the shows flowing, haven't you, Vincent, from your road shows?
No, TWiM has been just once a month because I don't have any road. But TWiV keeps going because, I have a lot of backup. So whenever we can't do something. But
I see.
I think we'll soon, I think in May we'll get to twice a month, hopefully.
Mm-hmm.
If not, certainly in June. And we will be Definitely. At ASM Microbe.
I probably won't.
And Elio will not. That's too bad. We'll miss you. But Michael and Michele and I will be doing a TWiM.
Oh, lucky you.
And we do have 196 other episodes that are online to listen to.
That's absolutely right.
You're selling our back catalog, Michele.
Yeah, they're all there, and they're there and archived, and you can listen to them. And Michele, correct me if I'm wrong. They're always going to be correct, right?
Well, we get letters. We get letters from our listeners. We're correcting us when we misspeak.
Yep. Yeah, the papers are, they're journal clubs, is what I say.
Yeah.
Paper findings can evolve, but that's the original finding, so it's all fine. Yeah, but we will try and get more frequency here. I want to tell everyone that at ASM Microbe, TWiM is going to be on Sunday, which is June 23rd at noon in the exhibit hall. And there are many other things going on at Microbe. Of course, that will be Michele's presiding meeting, right, Michele?
That's right.
You have a president's forum.
I miss.
That should be cool.
Well, it may be streamed live. You never know.
Some parts of it are, right?
Yeah.
But they do stream it within the place, so if you don't get in the main room, you could sit somewhere else.
Yeah.
I like that, actually.
So that's noon Pacific Standard Time then, correct?
Yeah,
yes.
Local time, yeah.
Mm-hmm.
And that's apparently okay with you, Michele, right? That fits in your schedule.
Yeah.
Because I did check your schedule.
I made it a priority.
And Michael and I, we can.
Well, Ray Ortega has found something for me to do, too, where I think they're going to play a version of What's That Microbe again, like we did last time,
year. Yeah. When is that going to be?
I think it's the Friday. Let me look at my calendar. I believe it's that Friday, the 21st.
Yeah, good. Very good.
So we're looking forward to that. Ada and I are going to do that again, and we're going to take questions from the audience, and they're going to play gotcha with us to see if what we don't know,
right.
which is a lot of things.
All right.
Yes.
And your partner Ada is Hagen.
And she is a graduate of the University of Michigan, and she's also a communications fellow for the American Society for Microbiology.
Mm-hmm.
And she writes a routine blog for the society that's a companion to our society's website.
Mm-hmm. And as everyone might have noticed, the ASM website has been
undergoing
revamped.
an evolution.
It's been revamped, and the new version is up, and should be a lot easier for you to find what you want. So check it out, asm.org. We thank them for their support. And now we have a couple of papers for you, and Elio's going to revisit something that we first talked about way back in TWiM number 21, an organism Paracatenula, right?
Paracatenula, a flatworm, marine flatworm, which has a most interesting symbiosis now with bacteria. Here's the story. Paracatenula lives in shallow waters. It's found in this case, off the island of Elba in Italy. I don't think this has anything to do with Napoleon having been exiled there, by the way. But this guy, Paracatenula, is only a few millimeters long, and it lacks both gut and mouth. It's not the only one. There's some other bugs, some other worms or beasts that do that. Notably, what is the name of it? Olavius algarvensis, which has no gut, no stomach, no anus, no nothing, but has 5 symbionts. This guy has only 1. Anyhow, what is funny about this is that this is, of all things, to me, a novel kind of symbiosis, because what the symbiont does is not only provide energy and nutrients like vitamins, amino acids, cofactors, and so forth, but also it stores them to deliver to the host when needed. Now, storage of nutrients is a new theme in symbiosis as far as I'm concerned. So I call this episode the Microbial Liver, because the liver is a storage organ for glycogen, and sure enough, glycogen is one of the compounds that these guys do. Are you with me?
Yeah.
This is a odd story. So what do? Bacteria acquire energy by oxidation of sulfur, namely reduced sulfur in the form of H2S, and they use oxygen as an electron acceptor. In other words, they do chemoautotrophy. No light is involved. In the process of burning H2S, they produce the compounds that the worm needs for biosynthesis. As I said, amino acids, vitamins, etc. Now, we discussed this, as I said, we discussed this before. In fact, if I make a little plug for our blog, there's an old paper there which goes with the one with the podcast we had. So if you Google Paracatenula and small things considered, you'll find it. Anyhow, so the arrangement of sulfur oxidation by symbiotic bacteria is not unusual. It is found, for instance, in the giant tube worms that are found near deep sea hydrothermal vents, which many readers must have, listeners must have heard about because it's very popular in National Geographic shows and so forth. But so it's not unusual for bacteria to use reduced sulfur, burn reduced sulfur to make energy and use that energy to fix carbon dioxide. But as I say, what's unusual is that these guys don't just make it and provide it to the host as such, but they store it in particular organelles, if you wish. So involved are energy-rich compounds, storage compounds such as glycogen, polyhydroxyalkanoates, excuse the long word, but this is a common polyalkanoates, is usually polybutyrate, which are used by many bacteria storage mechanisms, and kind of, a little bit oddly, a sugar called a sugar trehalose, which is also known to be a storage mechanism. And they deliver them by a specialized mechanism, which I'll tell you about in a minute.
But, and have you told us who's doing this beautiful work?
And thank you. It's a bunch of authors by the name of Jekle, See, Tetian, Leich, Liebeke, Kleiner, Berg, and Gruber-Wodicka. And they are mainly from the Max Planck Institute for Marine Biology in Bremen, which, by the way, must be quite a place because a number of wonderful publications have come from there. Plus, University of Calgary in Canada and North Carolina State, and a university in Paris, Université Pierre et Marie Curie. I got that out all right. Anyhow.
And they published this in PNAS.
This is a paper in PNAS, which is where it belongs. It's a very fine paper. I'm surprised. In fact, it's an extraordinary exhaustive paper. And so for people who like to read in detail about what one can study of such symbiotic relationships, this paper is a masterful presentation. It's really full of good stuff.
And it gives you a review of the physiology that this remarkable symbiotic relationship is going to actually use and transform to do some of the really neat biology that you're going to tell us about.
Amen. It's a really beautiful presentation of intermediary metabolism and energy metabolism. It has a lovely schematic diagram of the metabolism of this bug. The bug, by the way, is called, Candidatus Riegeria Sant'Andrea because of the Sant'Andrea Bay in the island of Elba. And it's called Candidatus because the bug cannot be cultivated. It never will be because it has a very reduced genome. It has a genome which is about 1.3 megabases, whereas the phylogenetic cousins it has have genomes of usually about 4 megabases. So reduced as it is, it's extraordinarily specific because it has in abundance the genes involved in glycolysis, TCA cycle, sulfur oxidation, and the Calvin cycle, that is the way to fix carbon chemosynthetically. So it has this diagram, I'm looking at it right now, it's just a gorgeous diagram. So this is quite a paper. Now, these bugs are alpha proteobacteria, which in itself is kind of unusual because most endosymbionts, this is an endosymbiont, it lives inside the host, and most endosymbionts are beta or gamma proteobacteria. That's only of interest to people who want to follow taxonomy of that sort. Now, the bugs are found in cells, specialized cells, called bacteriocytes, which are contained in an organ which occupies most of the body of the bug. In fact, half the body of the bug is bacteria. This is not much different than some other gutless worms, like the ones that I mentioned, but it's really quite remarkable. Now, that historically that sulfur metabolism is involved was suggested by showing that the worms have an abundance of white granules which turn out to be sulfur, and they amount to, in some cases, 20% of the body mass. So where does this sulfur come from? This is oxidized sulfur in the form of molecular sulfur, and it comes from the oxidation of H2S.
It's their waste product. It's their waste product.
It's what happens when you oxidize H2S. You make sulfates, and in turn, it becomes sulfur. Okay, so the paper has detailed and highly sensitive analysis through genomics, transcriptomics, and proteomics. And it tells you that these bugs in the genome have the genes for, as I said, sulfur oxidation, carbon fixation, etc. They are highly expressed, and the bacteria are very active in their chemoautotrophy. They do something which is not usually seen in symbiosis study. They also demonstrate that the biochemical activity is actually in the symbionts and not in the host by doing radioautograph of radiography of sections of the worms after giving them a suitable radioactive precursor. This demonstrates, since the radioactivity is found on the symbiont and not on the host, that this really the activity is by the host. As I say, you don't often see this done. It is confirming the strong suspicion, but it's nice to see that. So what can I tell you? The important thing here is that the material is stored inside the symbiont, and so how is it delivered? How does it come up? How is it given to the host? So they thought, well, like other symbionts, maybe the symbiont is just digested. Okay, that's done in other places where the symbiont, when it breaks up, when it's lysed, provides the host with its nutrients. Not so. These worms do not have a lysozyme, and apparently there's no disruption of the symbiont. Instead, and this is, we're going to talk about this in our second paper, they do it by a specialized mechanism, which is the production of outer membrane vesicles. That's not what the second paper is about, but it's something, in a sense, analogous. So outer membrane vesicles. Many bacteria produce these vesicles which are spewed out into the medium. Often they carry toxins. They're very important in metabolism as in pathogenesis. But here they are, these bacteria, I think so unique among symbionts, at least to date. They produce membrane vesicles which are loaded with foodstuff, and they can be digested by the host.
It's literally a little lunchbox for the worm. They're literally sending them lunch.
That's it. That's exactly right. So this is an efficient mechanism, and it provided the host with its needed nutrients. One other problem which exists here is that notice that in order to carry this out, you need both H2S and oxygen, and they don't coexist at the bottom of the sea. If you look at sediments, the lower layers of sediments have the H2S, and the higher up ones are exposed to oxygen, which is in the water. So how does this work? Well, apparently the worm provides an appropriate mechanism by traveling up and down, a little bit like a duck would. The duck goes down to the bottom of the water to feed and comes up to get air. So these are sort of duck-like worms. They go down to get the H2S. They come up again to get the oxygen. That's not an unheard of mechanism. The bacteria which do this inside the tube, but that's another story.
But it's very similar to what the editor of this paper, Margaret McFall-Nye, her squids effectively do this. They go up and down based on the light cycle, feeding on the various microbes and ejecting them. So it was a very cool behavior that the worm modulates in order to facilitate the metabolism of its symbiote.
That's right. So anyhow, this is the story. I think the amazing thing here is that this is a symbiosis which carries out a storage mechanism, has a storage mechanism to provide the host with foodstuff that way. It doesn't simply make it and spew it out. I think this is unique. Probably many other cases will be found. I imagine this is not the end of it. But here you have it, a microbial liver.
but a liver that occupies most of the flatworm.
That's right.
There's not one or two of these symbionts. The cytoplasm is packed with them. Figure 4, I believe it is, has got a lovely color image, so you can see the configuration.
That's right.
So it really brings up the question: who's controlling who?
Because also the question is, what's in it for the bacteria?
Right.
You can see what's in it for the host. This is a good question. I'm glad you brought it up. What's in it for the host is obvious, food stuff. What's in it for the bacteria? Well, the bacteria thrive there. So
it's the oxygen. It's the oxygen. Remember, the bacteria don't have the ability to go up and down any longer. They're obligate symbiotes. Yeah, and so the worm is effectively the transport vehicle for the critical limiting resources, which are the reduced sulfur compound and the oxygen, because you can't do one without the other. And so it's a trade-off situation between the two of them.
A beautiful partnership.
Yeah.
I wonder if the up and down of the worm is somehow controlled by the bacteria.
It could be.
Right?
Oh, boy.
I mean, the way I look at this is that the bacteria are running everything, and the worm is just like a bus for them.
A bus or an elevator.
And the bacteria are in charge. The worm is a passive person who happens to get a little food, but the bacteria need to provide food so the worm stays alive. Of course,
it's literally being thrown crumbs.
It's really remarkable.
So are the bacteria passed vertically to the flatworm progeny?
Interestingly, no, they're not. They're required at each generation. That's very odd. And again, I'm glad you mentioned that. I forgot to say that. It's somewhat unusual, but it happens. It happens in the squid that McNighfall studies. The guy studies, the one that Michael just mentioned. But in many cases, symbiosis is passed on from mother to child, as it were. But here it's acquired. So these bugs must exist in the water.
But how do they get into a flatworm that has no mouth or anus?
Aha! There's a question. Wonderful question. I don't know. Take it in.
I don't know either.
Take it in. When we find out, we'll let you know.
Maybe some kind of endocytosis.
That could be.
Yeah, could be. Maybe the worm don't have a gut, but that doesn't mean they don't have the ability to take up particles.
Right. Very cool.
Yeah, it's amazing. As I say, it's quite a study. If you want to know the details of this kind of metabolism and this kind of interactions, it's really loaded.
I have to thank Nathan over on Twitter. He's SciGuy999 for originally suggesting that we talk about this paper. Thanks, Nathan.
Great.
Yeah, it's good. But really, who's in charge here? That's great.
Could be the title. It could be the title of the show, because the next paper is going to really go back to telling us who's in charge.
Well, that's a great segue, Michael. Well, let's hear about our next paper from Michele.
And we're going to stick with eating as a theme.
That's right.
The title is Pathogenic E. coli Extracts Nutrients from Infected Host Cells Utilizing Injectisome Components. It was published recently in Cell, and it's from a group at the Hebrew University of Jerusalem, 2 different research labs collaborating, and the authors are Ratish Ranjan Paul, Amit Badya, Gideon Marno, Sarab Bhattacharya, Yaakov Sokol, Simi Kobu, Nami Katasowicz, Sigal Ben Yehuda, and Yehuda.
Yeah.
Say it again.
Yehuda. Ben Yehuda.
Yehuda. And Elon Rosenshine. And it is about a remarkable machine that equips many, many bacteria, gram-negative and gram-positive, to not only inject bacterial proteins into the cytoplasm of a host cell to manipulate its behavior, but also to extract nutrients. So it rests on appendage called a type 3 secretion system. And these really kind of burst onto the scene back in 1995, I believe. There was a whole series of papers from different labs identifying these syringe-like structures on the surface of many different pathogenic gram-negative bacteria. And through decades of research, we've learned that these structures evolved from flagellum, which are also large appendages on the extracellular membrane, extending from the extracellular membrane of gram-negative and gram-positive bacteria. But of course, they just make a motility organelle that allows bacteria to swim. But what we've learned is that these type 3 secretion systems, they're composed of more than 20 different components, and all of those components are encoded adjacent to one another on a piece of DNA that in many bacteria is mobile, which means the whole apparatus, all the genes that encode the apparatus can spread from one bacterium to another by horizontal gene transfer. So this is basically like one-step evolution, and it has had a big impact on the emergence of many different pathogens. So when I first learned about type 3 secretion, I definitely associated with pathogens. But what we're going to see here is that this really is a much more ancient mechanism, and one of its earlier functions appears to be extracting nutrients. So in particular, we're going to talk about 5 core components of these type 3 secretion systems and look at their capacity to extract nutrients from the host cytosol. And the model system that they're using is enteropathogenic E. coli, also known as EPEC. This is a human pathogen that can tightly adhere to the epithelial cells in our gut and form these spectacular pedestal attaching and effacing lesions, we call them. And from there, they can colonize the gut and either exist asymptomatically or cause mild diarrhea or quite serious disease, depending on the bacterial strain, the host, etc. So the question here is not how do they cause disease, but rather, once the bacteria have managed to adhere and colonize in the intestinal epithelium, how do they get nutrients? How do they get nutrients they need so that one bacteria can become 2, 2 become 4, etc.? So their first experiment.
I was just going to interject. That's a big deal because if you think about it, a novel pathogen coming into an established community, it's pretty hard to get to the food trough to obtain nutrients from the host because your intestine is already chock-a-block full of normal flora. And so consequently, it has to have some novel way of being able to get its nutrients. And that's
to outcompete.
Outcompete, yes.
Right, to outcompete all the others. Yeah, much like when I grew up in a family of 6 children.
That's right. You had to fight your brothers for the food.
Yeah. So the first experiment they did used really clever genetics, and that's going to be a theme throughout this paper, to ask if a particular bacterial cell can obtain nutrients. They started with a strain that was missing a gene that allowed them to make proline. So they're oxytrophic for the amino acid proline. They added to that proline oxytroph a fluorescent reporter of protein synthesis. And in particular, this is the green fluorescent protein gene that's being expressed from a promoter that is turned on when you add a small molecule IPTG. Okay, so they have this strain. It cannot grow unless it acquires proline from elsewhere, and they can ask if it's alive and making proteins by adding IPTG. So the experiment is they take an epithelial cell line, HeLa cells, and they add to it these proline oxytroph EPECs in rich media. So that allows these proline oxytrophs to attach, form these beautiful attachment organelles. Then they wash away the rich media and they add media that doesn't have any amino acids. And they also add IPTG to turn on that green fluorescent protein reporter. So in this way, they can ask, are these proline oxytrophs that are added to the HeLa cell cultures able to get the proline that they need and make GFP? So after doing this starvation, they then separate all the attached proline oxytrophs, ones that were attached to the HeLa cells, and separated them from the oxytrophs that were free, swimming, buzzing around in the media. And then just asked who's fluorescent and who isn't. So this is shown in figure 1b and figure 1d. And what you see beautifully is that the proline mutants when they are attached to host cells, they are able to turn on the green fluorescent protein, but the cells, the bacteria that are not attached, cannot turn it on. So we're having a correlation here with both the proline mutant and the wild type between attachment and the capacity to bypass that proline auxotrophy and make proteins, including GFP. So that was the first observation. They next asked if the amino acid proline and others were coming from inside the host cell. So the way they did that experiment was they first took their HeLa epithelial cells and they preloaded the cytoplasm with a small fluorescent molecule. And this was using some clever chemistry, a probe called calcinein that gets trapped once it's across the plasma membrane and in the host cell. So imagine they have these HeLa cells that are now glowing, their cytoplasm is glowing with this small fluorescent reporter. And then they repeated their infection experiment and either then starved, added tissue culture media that lacked amino acids or had them, and then again waited and then separated all the bacteria that had been bound to the HeLa cells and distinguished those from the cells that were unbound, and then just looked. Were the cells able to take up the fluorescent marker from the host cytoplasm or not? And that's shown in fluorescent images in figure 1F, and the numbers are shown in figure 1G. And remarkably, the bacteria that were bound became fluorescent, so that you can deduce then that they picked up this small fluorescent reporter that had been in the cytoplasm of the host epithelial cell. And it in some way traveled into the bacterial cell. So they know they're on to something, and then they want to ask more rigorously, do we have evidence that the auxotrophs are starving and are able to relieve that starvation by acquiring nutrients from the host cytoplasm? So in this case, they took advantage of a well-studied starvation response called the Stringent Response. And in particular, they used a promoter that they know is shut off when nutrients are plentiful and turned on when the bacteria, I have that backwards, turned off when the bacteria are starving, but turned on when they're not. And again, they hooked this Stringent Response reporter to green fluorescent protein.
And
so
the Stringent Response tells them it's true starvation, because that's been the hallmark in physiology for quite some time, that we know that when the microbe does turn on the Stringent Response, it's running out of something.
That's right. So they took advantage of that wealth of knowledge about the Stringent Response to make this reporter of starvation. So again, if you're green, you're not starved. If you're not fluorescent, you are starving. So they then
some of this knowledge about this Stringent Response comes from work by Michele, by the way.
Well, after a lot of beautiful work done by Fred Neidhart and Michael Cashel, and many others. But thank you. So they then took these Stringent Response reporter EPEC, and again infected their HeLa cells in rich media, then replaced it with starvation media, and then took a time series looking to see if the bacteria were going to turn on this GFP saying, I'm not starving. And this is shown in figure D, and it's really a beautiful time series, and it allowed the authors to use a sentence that I aspire to use. They write, the results were unambiguous. And I think you'll agree. They show photos at 0, 10 seconds, 20 seconds, 30 seconds, and so forth. And what you see is the attached bacteria go from dark to slowly becoming more and more bright green. So clearly showing that once the bacteria have attached, their starvation has been relieved. Somehow they're getting the amino acids that they need. They do another even more sophisticated twist on this experiment. They want to know if the process of achieving nutrients is dynamic. So they allow the cells to extract nutrients from their host HeLa cell, and then they go in with a laser and they bleach the green fluorescent protein so it's black again, and then watch. And sure enough, it recovers. The GFP fluorescence recovers. So this says the bacteria are continuing to acquire the nutrients they need while they're attached to the HeLa cell. So this is getting really, really beautiful and a nice combination of immunofluorescence, or fluorescence microscopy, and some bacterial genetics. They then wanted to know, is this a trait shared by other pathogenic E. coli, or is it just our strain? So they looked at a couple other enteropathogenic E. coli strains. They saw the same thing, but when they took kind of garden variety lab strain of E. coli K12, it did not extract nutrients from host cells, even when they had engineered the K12 to tightly adhere to the HeLa cell using invasin, a well-known virulence factor that equips bacteria to tightly adhere to host cells.
Say that again. I didn't understand what the...
They were curious whether it was adhesion per se. Do they just need to be totally associated with the host cell, or is it something special about the organelle that is mediating that attachment?
Mm-hmm.
If I could interject here,
please. This
was one of the more exciting days in the lab of the first offer, Dr. Paul. He writes that there were many times he and his supervisors and colleagues were really excited about this finding of microbes, principally this E. coli, being able to take nutrients out of the cell, out of the host cell, and be able to see it with this green fluorescence protein. But this was gaining access to the nutrients during infection was the most exciting part. And Michele is going to take us in this next set of experiments, she's going to take us to what was his year-long adventure trying to figure out how this organism was able to effectively make this pipe that was siphoning nutrients out of HeLa cells, delivering it to this bacterium. And it's this year-long excitement that he found that was so exciting for him and his research colleagues doing this experiment. Michele, now take us to the fun stuff.
Will do. So they next took advantage of, again, decades of really beautiful structure function studies and cellular microbiology, where we've learned particular components that equip EPEC to form these attaching and effacing lesions and colonize the intestinal epithelium. So, for example, they were able to create an EPEC strain that lacked the adhesin intimin. They found that still acquired nutrients from the host. They made a mutant that lacked the bundle-forming pilus, this other very impressive attachment organelle. Nope, you don't need that. You can still extract nutrients without the bundle-forming pilus. Then they deleted the pathogenicity island called Li, the locus of an effacing.
Anaeroeffacing.
The Li locus.
Anaeroeffacing.
There it is.
Yes.
This is a mobile genetic element. It encodes more than 40 genes. When they deleted that entire element, the EPEC was no longer able to extract nutrients from its host. And if they took the Li element and transferred it into garden variety E. coli K12, now K12 acquired the capacity to extract nutrients. So this is a classic genetic demonstration that the Li element is both necessary and sufficient for E. coli to extract nutrients from a host cell.
Another unambiguous result.
Yeah. And again, the images are shown in figure 3. It's really, really beautiful to look at. And they also do quantitative work. So again, because these type 3 secretion systems have been studied for decades, we know a lot about the components, the ultrastructure, what pieces do which. So that is actually summarized for us in a schematic, figure 4A. We can see just how many genes are involved and which genes contribute to the assembly of which components of this molecular syringe. And in this case, we're going to focus on what they term the core genes. These are 5 genes called ESC, R, S, T, U, and V. What they do is create an EPEC strain that lacks the entire Li island, but does encode these 5 core genes. And they find, yes, it can extract nutrients from the host cell. If they delete the 5 core genes, no, it cannot. Now, if they can genetically complement that defect by providing only the 5 core genes on a plasmid that restores nutrient extraction capability. And they can take just the 5 core genes on a plasmid, put it into garden variety E. coli K12, and now it gains the capacity to extract nutrients from host cells. So Eureka, they've got it now defined as this small intermembrane protein complex that is both necessary and sufficient to extract nutrients. So in the next series of experiments, they turn to the morphology. How is this actually working? So for this, they turn to very sensitive imaging called extremely high-resolution scanning electron microscopy. This is shown in Figure 5, especially I think it's panel C, or panel D, subpanel C. And what you see are these really striking protrusions from the outer membrane. So they're a little bit spiky. To learn whether they are made of membrane or protein, they use another fluorescent dye. This one happens to be called FM464, which basically can intercalate into greasy membranes. And indeed, these protrusions do stain with the membrane dye, and the membrane protrusions, these needle-like projections, are decorated also with antibody that stains lipopolysaccharide, abundant component of the outer membrane of gram-negative bacteria. So by those 2 criteria, they say we need the 5 core genes to make these membrane fingers, and they are, in fact, outer membrane extensions. What was exciting about this is it connected their observations to earlier work that the Ben Juhada lab had published in a series of really beautiful papers. Starting in 2011, they showed that Bacillus subtilis also makes these membrane nanotubules, they call them. And in that case, Bacillus uses them to communicate with other bacteria. So I want to really give credit to the work that had been done previously in Bacillus subtilis, because those of us in the pathogenesis field, when we discover something new, we think, oh, our pathogen is such a terrible scourge of humans because it has this special capability. But in fact, we're going to see that this was an ancient function that is now widespread in the microbial world, and it's not something special that only pathogens can do. So we'll return to that in the end. So the next experiment they did then was they wanted to really be rigorous and make sure that those tubules that they saw were the conduit for the nutrients, rather than some indirect mechanism. For example, perhaps those membrane projections are killing or poking a hole in the host cell, and the nutrients are just leaking out to the vicinity. And if so, cells that can't make these membrane protrusions perhaps can cheat and just...
Are they like a mosquito biting somebody?
Yes,
right.
And where you're going to have bleeding.
A neighbor could cheat and take the nutrients that the core complex had liberated. So to ask that question, they again did a very elegant experiment. They had a bacterial EPEC strain that could make these nanotubules, and then they had a mutant that lacked the core genes, and so it could not. They made a 1-to-1 mixture and then infected their HeLa cells, and each of the the wild type and the core mutant were labeled fluorescently a different color. So they were either green or they were red. So they could distinguish the core positive from the core mutant bacteria. So when they added a 1-to-1 mixture to HeLa cells, they then searched the cells, the fields, for cells where there were host cells where there was a mutant bacteria, a green one right next to a red one, and asked, can the core minus mutant cheat and acquire nutrients? And figure 6 C shows images and B shows the numbers, that no, you cannot cheat. This doesn't work in trans. Only the cell that is able to make this membranous protrusion made up of core components is able to acquire nutrients. So they've learned something more about what the mechanism is, and you can deduce that the nutrients are flowing through the pore made by this nanotubule. Now, the next question they asked is, since type 3 secretion systems that have this core complex are really best known for exporting virulence factors into the host, and yet here we're finding that nutrients are flowing in the opposite direction, can one cell do both? Or as a population is dividing, are there two different subpopulations, one that is doing the virulence export and the other one that is acquiring nutrients? So to ask that fairly sophisticated question, they use single cell analysis using a strain that has two different reporters. One that will become red if it is acquiring nutrients by relieving the stringent response, and the other that will turn green if it is injecting virulence factors through its type 3 secretion system.
I wasn't quite sure how that works. How does it tell you that?
Well, what they do, and we can look at figure C6D, is they ask if they only see green cells, or do they only see red cells, or do they see some that are expressing both fluorescent markers and therefore turn up as yellow in the green-red overlay. So, for example, in panel D of figure 6, by phase contrast, we can see the phase dark bacteria. We can see which ones have expressed the type 3 secretion effector. We can see which ones are not starving. They don't have a stringent response, so they're bright red. And we can locate in the population some that apparently have done both. So they don't have evidence that the cell can only do one or not the other. It looks as though one bacterial cell is able to do both export through type 3 secretion and import through this host nutrient extraction mechanism. Now, the resolution, of course, is not good enough to see if you've got two-way traffic in one nanotube, or whether there's a traffic light that lets traffic go one way for a while and then switches and reverses flow. So they admit there's still a lot of mechanistic details to be worked out, but to a first approximation, it looks like any one cell in the population can do both export and import.
Does that make sense? It makes sense that it would have to do both, because the first rule is you can't violate the hydrogen tight membrane or the bacterial cell will be dead. So you have to maintain membrane integrity. And by virtue of the fact that you maintain membrane integrity, you would anticipate that unless there is a gatekeeper that is excluding the nutrients from the host cell, there would be no way to get around the laws of thermodynamics, because diffusion kinetics are going to drive the nutrients from the host into the bacterial cell, because recall that the proteins that are exported via type 3 secretion are effectively ratcheted out through the ATPase function of the syringe mechanism. And for the nutrients to come back in, you have thermodynamics driving it, where you have a tremendous driving force of the solutes wanting to go to a lower concentration. But in the bacterial cell, you have the gate closed, and only the proteins that are in that secretion mechanism can go out. But it must be a sort of like our veins, where the valve allows flow the other way into the bacterial cell.
Makes sense to me.
Yeah.
So they next want to see just how widespread is this phenomenon. And recall that the type 3 secretion system syringes that we've talked about initially are known to be evolved from flagellar apparatuses that mediate flagellar-based motility. And these 5 core components that we've focused on here are shared. So the flagellar apparatus still also contains these same five core components. So this put them in a position to do really rigorous cross-species complementation tests to ask whether this nutrient extraction function has been retained by the 5 core components across evolutionary time. So the way they do this is they have their EPEC mutant that lacks the 5 core genes, so it cannot extract nutrients. They then show that they can complement that if they use the 5 core genes from Salmonella typhimurium, pathogenicity island 1, or pathogenicity island 2. So Salmonella has 2 different type 3 secretion systems. Its 5 core components can substitute for the EPEC core components. Not only that, but the 5 core components from EPEC flagella can also substitute for the nutrient acquisition activity. And so can the 5 core components from the flagellar apparatus of Bacillus subtilis, which is a gram-positive bacterium that lives in the soil. So this and other bioinformatic data allows them to say that this is really an ancient function of this microbial appendage whose core components are still conserved and very widespread in the microbial world. And with another paper that this group, Ben Yahada's group, published a month earlier in Cell Reports, they're able to summarize this study and their other companion paper with the really lovely statement that core-derived nanotube is a ubiquitous organelle that facilitates intercellular molecular trade across the bacterial kingdom.
Mm-hmm. Wow.
So it's
That's
not
a profound
just
statement.
pathogens. Yeah, but it's well supported by beautiful fluorescence microscopy, genetics, and quantitative data that is in this paper, their companion Cell Reports paper from March of 2019, and then a series of papers that led to this, starting with some work by this group in 2011. So a great collaboration between these two groups at the Hebrew University has really changed how I think, and we should all think about type 3 secretion. It's not just delivering virulence factors, but also likely equipping bacteria to extract nutrients from the host cytoplasm.
So what's the effect on the HeLa cell in these experiments? Are they okay?
It's leaking. It's feeding the bacterium.
I know, but do they remain
Can
viable?
They keep up?
Do they live?
Yeah.
Do they die? What?
So in colonization assays in mice, but also in patients, people can be colonized asymptomatically with EPEC for long periods of time, so without symptoms. So presumably they can extract just enough nutrients from the epithelial cells without doing damage.
So in a real life situation, do you think the bacterium remain attached to the cells for long periods with these tubes in them and these intravenous tubes?
Yeah.
And just live there and then they divide, and then the other cells attach and do all of this. So it's not a dynamic situation where it attaches, detaches, and so forth.
Yeah, that's a good question. I don't know if people have done kind of longitudinal studies in patients to be able to ask whether it's the same population that is there. But I think that data is available. I think people can be chronically asymptomatically carrying the bacteria at low levels. Presumably, at some point they detach and are spread through fecal oral route to another unsuspecting host.
So when they detach, then the core complex closes off, right, on the bacterium, so it doesn't leak out.
Presumably.
Of the bacterium.
Yeah. Good questions.
You can imagine that, at some point the bacterium is going to think, why don't I just go inside this cell?
Well, that does indeed happen.
Yes.
That does indeed happen.
Yeah.
Right.
And Well, Dr. Paul is an Indian transplant to Israel. And he writes in his response to Michele's query to get some background information on him is that he was trained principally in India. He obtained a master's in genetics from the University of Calcutta and his PhD from the CSIR, the Indian Institute of Chemical Biology. And unfortunately, his fellowship research in India did not provide enough earning potential for himself because he needed to support his wife, his son, and his widowed mother. And so since he was adamant to do high-quality research, he moved to Israel in 2014 for a postdoc while his family stayed in Kolkata, India. And his family supports him in every aspect along the way with his career. Outside of work, he's a competitive table tennis player, and he's represented the Indian Institute of Chemical Biology as a captain in some inter-institute tournaments. And he likes traveling. Whenever he gets to travel, he travels to different places in India and Israel. And as I alluded to during Michele's overview of his paper, he's had some really exciting times during the course of this particular study. He and his colleague Amit Bhatia, they saw those high-resolution SEMs with the wild-type EPEC forming this membranous nanotubular connections, but the core mutants were unable to do that. And that was their aha moment. And that, in concert with this other observation where Amit and Sarbra found the nanotube formation in the bacillus, was really, if you will, headline news. And again, the immune SEM that the EPEC is connected to the host cell through these nanotubes is really quite remarkable. And he also gives credit to his lab members that have helped him throughout the course of the study, not only by helping with experiments, but also in their kibitzing, their valuable ideas by showing confidence in his interpretations. And he wants to particularly call attention about Amit and Yakov Sokol. Without the expertise of Amit and the SEM, which, as everyone knows, SEM is really quite artful, and the finding of those nanotubes would not have been possible. And Yakov Sokol did the initial assays with that first experiment with that fluorescent molecule that was inside the HeLa cells, that the HeLa cell has the esterase that cleaves off the molecule, making it fluoresce once it gets into the bacterium. And the bacteria lack that esterase. So the only way the calcium can fluoresce is if it was first inside the HeLa cell. The HeLa cell did the dirty deed, clipped it, and then transferred the calcium into the E. coli cell. And that needs the direct contact between the bacteria and the cell. And that's how all of this started, with the very clever genetics. And one final thought he wants to leave for his junior colleagues, but I think it's really for all of our colleagues in general, is that the key to success in research is patience and enjoying the work. The hypotheses may fail, but an analysis of the reasons of the failure patiently will lead to the fact of the phenomenon. And I think that was quite apparent here, and it's really an elegant piece of work.
Very good.
Michele, is this the first time these structures have been shown to extract material from a eukaryotic cell to a bacterium?
I think so, but I haven't. I'll admit that I haven't gone back and read like the Bacillus paper that they published in 2011 showing communication. Right. And then their recent Cell Reports paper it looks like it's also focused on the biogenesis of these nanotubes, but I don't know whether this was the first assays looking specifically at nutrient extraction. Mm-hmm. Certainly for a pathogen, this would be the first. And it's this wonderful collaboration between the Sonnenschein group and the Rosenschein group, and the Ben Yehuda group at Hebrew University that's led to this series of really beautiful papers.
Mm. All right. Thank you, Michele.
Yeah.
Yes, that's cool. Very cool paper. And you're right. They both are about food, about eating anyway.
Right. And it's what we teach the medical students. The goal of every microbe is to become 2 microbes.
Yes.
Nothing more complicated than that.
It's really quite simple.
You need food to do it.
So intercellular microbial trade could apply to both papers today, right?
Yes, absolutely.
I think so.
All right. Let's, we have just a couple of emails here I'd like to go through. First is from Anthony. He writes, anti-vax and now anti-wash. The biggest advance in public health was the introduction of cotton clothing that could be vigorously washed. Well, I would say, Anthony, the biggest advance is the toilet.
I think so, too.
Maybe cotton clothing would be somewhere up there. It's strange to see a clothing manufacturer seeking to undo this, and he sends a link to a CNN article. It's a interview with the CEO of Levi's, who says he has some tips for jean owners. Don't freeze your jeans and don't put them in the washing machine. He turned heads and noses at a May 2014 event when he announced that he had never washed the pair of jeans he was wearing. He still hasn't washed that now 10-year-old pair of jeans, he admitted on Thursday.
Does he take them to the dry cleaner?
How's his social life?
I think he takes them to the dry cleaner.
Don't freeze them and don't put them in the washing machine. He says freezing doesn't work. I don't know what.
No, definitely freezing does not work because we all have -80s full of frozen bacteria.
That's right.
We know that.
Yep.
We know freezing won't kill off all the microbes because, especially if I have 2 words for him, stool happens.
Yep.
And
maybe he lives in sunny San Diego and he can just use the natural UV
and air them
sterilization from the sun.
And air them out. Yeah, you never know. But this whole issue about not vaccinating children and not washing because you're afraid you're going to wash off good bacteria. Well, you will wash off good bacteria, but they just make more. You're not going to jumping in the shower doesn't make you sterile. It effectively just debulks.
Yep.
Debulks your body mass. Debulking is good. Debulking is good. So
we have one from Ben who writes, "Dear hosts of Fusobacterium nuclei TWiM."
Oh, it's clever.
Sorry, I'm stretching it a little there. I've been listening to the Twix series for a few months now, starting with TWiP because I'm doing a parasitology PhD, but now working my way back through the TWiM catalog. I have a suggestion for a recent paper on bioRxiv where the authors are able to individually identify 19 different species of bacterial pathogens using 3D tomography and bacterial learning. It gives a link to that article. While it hasn't been peer-reviewed, I think the fact that people are coming up with these kinds of projects now and they actually work is incredible. I also heard a talk a little while ago about someone who had developed a flow cytometry assay based on machine learning that could distinguish between a number of bacterial species and determine their antibody sensitivities within only 30 minutes or so, the idea being it would be done while you wait at the doctor's office. This sounds like the Poliano's
right.
Yes.
approach to antibiotic sensitivity, right?
They use it cell sorters.
Yes. So that's what he might be referring to, and not antibody sensitivity, but antibiotic sensitivity. Does that make sense?
That makes more sense than the antibodies.
I was interested in this paper mostly because a part of my PhD is to develop automated and quantitative super resolution microscopy tools for studying protein function in malaria parasites. A few months ago, I wrote up a grant to try and quantify live malaria parasite invasion into erythrocytes using tomography and machine learning, just like the paper, because we can differentiate the extracellular parasite an uninfected cell in each stage of the intracellular life cycle based on differences in refractive index. Unfortunately, it wasn't successful, but that won't stop me from trying again. While doing a parasitology PhD, I'm fascinated by microbiology as a whole. This no doubt is fostered in part by the fact that until a few weeks ago, our lab was housed inside a bacteriology lab. Because of that, one of my co-supervisors is Professor James Patton, who works on the pneumococcus and has some very cool projects looking at the relationship between sugar metabolism, invasive disease, and also serotype-independent vaccines. Even though we are well and truly into autumn, it was a beautiful day today in sunny South Australia at 31 degrees Celsius.
That's warm.
If it keeps going this way, I don't think there will be many leaves to clean from the streets. Sorry for the lengthy email, but thank you all endlessly for colonizing my ears with microbial magic.
That's
Thank
nice.
you, Ben.
Ben is a student in University of Adelaide. Hope I don't say it right, but you get the idea.
Right.
Richard sends us a paper, more to substantiate multiple oral microbes found in periodontal disease lesions as possible causal factors for Alzheimer's, complementing the study reviewed in TWiM 195. And this is a paper to review periodontal herpes viruses, prevalence, pathogenicity, and systemic risk. So, Michael, you and I did with P. gingivalis, right?
Yeah, Porphyromonas gingivalis.
And so it's implicated in Alzheimer's, and so are herpes viruses. And here's one about oral herpes viruses. Yeah, that's where they can go. Justin sends us a link to an article. There are fungi all over the space station, and now we know what they are. This is an article in space.com by Elizabeth Howell, and they have looked at the fungi growing up on the space station. And
you don't want athlete's foot on the space station.
They collected samples.
They don't wear any shoes. They're always in socks.
That's right. Over 14 months, they collected samples from locations on the International Space Station, and they looked at the fungal diversity. All right, there you go. You might as well. And they have a plate here. A lot of microorganisms found on busy public spaces on Earth. How about that? And one more from Ryan. I just read a few articles on gut bacteria and autism connections. Right now, I think the connections still need to be verified. I don't know how many years will the FDA or CDC need to confirm this or not. Here's a paper, and it's a review of a paper in Science News. Autism symptoms reduced nearly 50% 2 years after fecal transplants. So, yes, this requires extraordinary proof, I think.
It does.
Well, and it's generating a lot of excitement, but you can imagine these experiments are difficult to do in a controlled fashion, and so I think the jury is still out.
Yeah,
and expensive. Yeah. That's true.
Well, it's hard to properly control them. You send people out into the world, and you don't know what's happening, right?
Right.
Well, it's going to be very similar to the Alzheimer's one, where you have the data in the paper that we reviewed looking at Porphyromonas was pretty convincing, but similarly, the data associated with the herpes virus are also pretty convincing. So it's probably not a one-hit event. So Alzheimer's, like autism, is probably multifactorial. That's dependent upon the genetics of the host, as well as the circumstance and epistatic lesions and all sorts of things.
And how the host responds.
Yes.
Yeah.
Here's the recommendation of Michael Pollan. 3 rules for eating. Eat well, eat food, not too much, mostly plants.
Yeah.
There you go.
That makes sense.
Okay.
All right.
And bacteria are still classified as plants as they originally were, right? We're going to get letters for that one.
They're classified as plants?
Originally, bacteriology was a subdiscipline of botany.
No, but no longer, of course.
No longer.
Yeah.
We digress.
Yes.
TWiM
197, you can find it on your favorite podcast player, whether it's on your phone or tablet. And please subscribe. It's free, and you get every episode, so you can listen to what you want. And if you really like what we do, consider supporting us. You can go over to microbe.tv/contribute. We have a few ways. where you could say, give us $1 a month. And for that, you don't get not only TWiM, 2 shows a month, but you get 2 shows of TWiP, you get a show of Immune, you get a show of TWiVO, and you get 4 shows of TWiV. What a deal. $1 for all that knowledge.
Wow.
You can't beat that. There's no better deal in the world.
For microbes.
And of course, if you want to send us an article or say hello, TWiM@microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
My pleasure. Great to be with you.
Thank you for joining us on your vacation. It's really nice of you. Elio Schaechter's at Small Things Considered. Thanks, Elio.
My pleasure.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And I am Vincent Racaniello. You can find me at virology.ws. I want to thank ASM for their support of TWiM, Ronald Jenkins for the music, and Ray Ortega for editing this episode of TWiM. Thanks for listening, everyone. See you next time on This Week in Microbiology.